Welcome to the next video, Why and How is there gastroesophageal cancer? In this video, we're going to be going back to Biology 101 to really get an understanding of the basic principles that are necessary to understand later videos when we're talking about molecular testing and targeted therapies. In previous videos, we talked about the what, the where, and the who's of gastroesophageal cancer. What is cancer? Where is gastroesophageal cancer first anatomically? The three major subtypes: esophageal squamous cell cancer, G junction, esophageal gastric junction adenocarcinomas, and gastric cancer non-cardia. And also where globally each of those three main subtypes tend to have higher incidences. We also looked at who's at risk for cancer, some of the known factors associated with each of those three subtypes of gastroesophageal cancer, and also known causes. We also looked under the microscope and saw that cancer cells look different to the pathologist compared to normal cells, and that they can readily identify where these cells originated in the tumor epithelium, the most inner layer of the stomach or esophagus, and how they invade downwards into the lower layers of the esophagus and stomach, and eventually can gain capability to travel distantly in the body. But why and how is there cancer is the focus of this video. And in order to understand that, we need to go back to the central dogma of biology. And that is that DNA is the blueprint of life. All living organisms follow this blueprint. And what is DNA? DNA is made up of two strands of DNA, which is a sequence of base pairs, only four possible base pairs: adenine, thymidine, guanine, and cytosine, or ATGC. And it's that specific sequence that's important that dictates what the organism is. Is it human or whatever? And humans share 99.9% same sequence. It's the 0.1% that makes us unique individuals. So each of those strands comes together and binds specifically through specific matches of these base pairs. So if your sequence on one strand of base pairs was ACGT, then the other strand would be complementary to that, because an A always binds to a T and a C always binds to a G. And similarly, if this was a G on this side, then this would be a C on that side. The DNA resides within the nucleus of a cell on chromosomes. And the next thing to understand is that there are 3 billion or so base pairs in the human genome. Anything having to do with the DNA is referred to the genome. And times 2, because we have one copy from mom and one copy from dad. We have, amongst all those billion base pairs, we have about 20,000 genes. Again, 20,000 from mom and another copy of this each gene from dad, so 40,000 genes. There's a lot of DNA that doesn't code for genes, that is just in between the genes. And so less than 1% of all the DNA that we have is actually coding for genes. And all of this DNA resides on 23 chromosomes times 2, one set of 23 from mom and another set of 23 from dad. And the next thing to understand in this central dogma is that each cell has the same copy of all of that DNA, each cell in our body. But certain cells will turn on some of those genes, and the way they do that is they transcribe the DNA into RNA. RNA is a single strand as opposed to a double strand, and that single strand is made from one of those two DNA templates. So if we were using this gray template, then the blue copy would be made. So what happens during transcription is that these two strands unravel and come apart. And if we're using the gray as the template, then the blue would be made. So if the gray template said C, G, T, etc., then the RNA would be being built with these building blocks. And if this was a C, this would become a G. If this was a G, this would become a C, etc. The other thing to note with mRNA is that T's become U's in RNA. And so this messenger RNA then travels from the nucleus where it was made into the cytoplasm, which is inside the cell still, but outside of the nucleus, to this Golgi apparatus in green. And in that Golgi apparatus, the messenger RNA is translated into protein. Protein is made up of building blocks that are called amino acids. And there are 20 possible amino acids that are put in sequence based on what the DNA code says. The next thing to understand is that the DNA is every 3 base pairs is called a codon. And that's because this codon, as I'll show you in the next slide, specifies what building block amino acid would go in that spot as you're building this protein in the Golgi apparatus through translation. So in this case, a GCA specifies that an alanine amino acid should go here, denoted with a green, etc. And so again, it's the specific sequence of these amino acids, which ultimately is dictated by the sequence of the base pairs in the DNA, is what is making this protein unique and doing its specific function. And so, as I was alluding to earlier, specific cell types will turn on through transcription and translation certain genes. Skin cells will turn on skin cell related genes. Stomach cells will turn on stomach cell related genes, etc. And it will turn them on and off appropriately as indicated in this normal equilibrium that is tightly maintained. The next thing to understand is that cells divide and they replicate themselves. And we do this as embryos, as we're growing and as we're babies and children. But even as an adult, we have to divide to replenish cells that are dying. Our bone marrow and blood cells often are turning over and we need to make more. And so through, again, tight regulation, cells are told to grow and divide and told when to turn off in a tightly regulated manner. Now, you can imagine that every time a cell divides, it has to replicate itself, a carbon copy of all of its DNA, and there's a lot of those base pairs. So if, for whatever reason, and we'll talk about it later, the sequence is changed, even in one location, this can then lead to a change downstream in the amino acid, and therefore a change in the protein and its function. And in this setting of cancer, oncogenic changes, carcinogenic changes in the DNA can lead to oncogenic changes in the protein. I mentioned that there are 20 building blocks of amino acids, and this table shows you all of those 20 amino acids that are listed here, and what the codons in the DNA and then the RNA are that specify which amino acid you're going to get. You will first notice that there are more than one codon that will encode a specific amino acid. So in this example, there are 4 different codons that would specify a phenylalanine, etc. And so there's some redundancy there. Regardless, this sequence, as it's reading from the mRNA, will then dictate and use building blocks to sequence the protein, and this sequence is what is important and dictates what the protein is and what it does. An example is a protein like an immunoglobulin or IgG, which is an antibody that is part of our immune system. It is a protein that is encoded by a gene, and you can see that this specific amino acid, where it's located in all the different locations on that particular protein, that's folded up on itself. Again, this folding is dictated by the exact sequence of this peptide. The last thing to show on this slide is that, alluding now and getting into why and how there could be cancer, is that if this codon is changed for whatever reason, so a UUA is changed, for example, to a UAA, one thing is changed, it can change from stipulating that it should be a leucine amino acid to a stop codon, which means the protein doesn't, the amino acid peptide stop adding more amino acids after that point. Another example of many is if this AUU codon, which is stipulating to use an isoleucine in the next position, is changed to an AAU, one change in one base pair, then you'll get instead of an isoleucine, you would get an asparagine amino acid. And again, these things matter, as I'll show you. Even just one change can alter the function of the protein. So how do we get these changes? Well, I was alluding to the fact earlier that every time a cell divides, it has to replicate all of those specific billions of base pairs in a specific order. And because of that, it is prone to having a mistake here or there. Now, thankfully, we have something called proofreading genes that make enzymes that go back and look at the sequence. And if it looks different, it can change it back. But even so, sometimes it gets through the proofreading system and a mistake can occur. And if such a mistake occurs through one's lifetime, through all the cell divisions, this would be an acquired mutation through one's lifetime. And that's referred to as a somatic alteration. One can also inherit a problem in the DNA sequence from one of your parents. And when you inherit a problem, it would be called germline. It was in every cell, not just the one cell that was dividing and made a mistake, but in every cell in your body, because it started from the very get-go, from the beginning. And so you can imagine that this could accelerate the carcinogenesis, the development of cancer, because you've already had a problem in a DNA from early on, as opposed to acquiring it through one's lifetime, often later in one's lifetime. An example of an inherited susceptibility is a problem in the proofreading gene that I just mentioned, that goes and proofreads and changes and corrects random mistakes through DNA replication. And so if you inherit a problem in your proofreading gene, then you would be at risk for multiple mistakes through one's lifetime. And again, a cancer usually early on in your lifetime. And an example that we're going to talk about in a later video is Lynch syndrome, or deficient mismatch repair syndrome, that occurs not uncommonly in gastric cancer. And then finally, the majority of cancers are accelerated by exposures through one's lifetime to various things that are insults to the DNA, that change and alter the DNA, infections, chronic inflammation, radiation, and mutagens, carcinogens like tobacco smoke, that will change the DNA sequence and, of course, then increase the risk of cancer. And this, because it's acquired through one's lifetime, again, is referred to as somatic. It's not uncommon to have combination of inherited problems as well as somatic problems that are acquired through one's lifetime. Now, there are four major types of genetic changes. And the first thing to get in our minds is to understand that there's two classes of genes. One are called oncogenes, which in a car model would be the gas pedal in the system, telling the car or the cell to grow. And there's tumor suppressors, which are the brakes in the system, telling the cell not to grow. And in cancer, through these various four major types of genetic alterations, you can get overactivation or constitutive activation, we say, of oncogenes, and loss of function of the brakes, the tumor suppressors. So these four major classes we'll talk about quickly. The first one are point mutations, which I was referring to in examples previously, where if you have a specific DNA code of base pairs, and for whatever reason, if the code is changed, say in this case it's changing from a G to an A, then that would change the codon, and that could change the amino acid, and that could change the function of the protein. If the point mutations can lead to either overactivation of oncogenes or loss of function of tumor suppressors. An example shown here is an overactivation of an oncogene called BRAF, with a change of a valine amino acid at the 600th position of the protein to a glutamate amino acid. And this change overactivates this oncogene and inappropriately always on, always telling the cell to grow. The next class of alterations are called copy number changes. And these copy number changes, remember, we get one copy of every gene from mom and one copy of every gene from dad. in every normal cell. In cancer cells, it's not uncommon to have multiple copies of an oncogene, and that leads to more RNA through transcription and more protein through translation and more activation of that oncogene inappropriately. And an example of that in gastroesophageal cancer, again, is HER2 gene amplification, or ERBB2, which occurs in about 10 to 15% of gastroesophageal cancer. A third alteration is called translocations or rearrangements or fusions. And if on the chromosome there's genes in sequence, genes, as I was talking about earlier, are turned on and off based on a promoter, which is this, depicted by this black box. And so certain cells will have some genes on all the time. Certain cells will turn a gene on and off less often, because it has a different function. If there's damage to the DNA and there's breakage in the DNA, and then splicing back in a different order, like in this example, this part of the DNA has gone here and this part of the chromosome has gone here, now you've created a fusion protein where this oncogene, which was normally regulated by this promoter, turned on and off sparingly and appropriately to when it needed to be on, now is being controlled by a different promoter. And if it is in front of a gene that's on all the time, now this gene is always there and always on and always active. And so this is not uncommon in cancers, and I'll show you that in the next slides. And then finally, you can get in small insertions or deletions of the base pairs into the gene that would then push all the other ones down. And so you have an extra A here, for example. So it's CTA and then GTTGC. So remember, the codons are important to stipulate what the next amino acid is. So if you add an extra base pair there, then everything will be shifted. We call this a frame shift mutation. And therefore, every amino acid downstream will likely be different and alter the function of that protein. Usually this leads to loss of function of that protein. In terms of these four major classes of genetic alterations, you can see the makeup of across cancers and their incidence. So the majority of changes in the DNA are base pair substitutions, those single changes in one base pair. But a close second are gene copy number changes, amplifications, or deletions of the gene. And this is pertinent, again, to gastroesophageal cancer with HER2 gene amplification. So going back to the central dogma of biology, where DNA is transcribed to RNA and translated to protein normally, if you get changes in the DNA, this can then lead to altered function of the proteins, either overactivation of oncogenes or loss of function of tumor suppressors. And again, usually it's a combination of these things in any given protein, where if you look at the genetics of a given cancer, there's often a few oncogenes activated and a few tumor suppressors inactivated. And ultimately, over time, as these changes occur and accumulate, you will then lead to a full-blown cancer cell that is always growing, always dividing, always being told to do so, and losing the capability of the brakes and stopping that phenomenon. So in this video, we talked about the central dogma of biology, that DNA is the blueprint of life, and that DNA is transcribed to RNA, which just translated to protein, and it's the sequence of the DNA base pairs that dictate the function of the protein, and changes in that DNA through various means, whether it's inherited or whether it's acquired through time, through exposure to various things in one's lifetime, or just through random copy mistakes, you can get oncogenic changes that lead to cancer. This is pertinent, and this understanding will be pertinent to molecular testing and understanding what we're looking for, how we look for it, and also targeted therapies, leveraging our understanding of these problems now with specific therapies that use these changes as an Achilles heel to target those cancer cells. In the next videos, we'll be looking at two specific mutations in genes, the first being CDH1 mutation, for diffuse type gastric cancer, which can be inherited or acquired, as well as changes in mismatch repair genes that can be inherited in the Lynch syndrome or acquired through one's lifetime, that can lead to a condition called microsatellite instability high, or MSI high. So stay tuned for that, and thank you for your attention.